We'll be discussing melanocortin-1 receptor and the mutation in which the arginine 67 is replaced with a cytosine 67, and that of course leads to lower pigmentation. This particular case was about beach mice, and it's very monumental work because before this case was studied, scientists believed that evolution had to happen in small steps, but this kind of proved that evolution could happen in more drastic steps and happen a little bit more quickly. Because with this case, we had beach mice who are pale versus their, land, inland counterparts who were darker, and not a lot of time had passed in between the two variations. So, how did that happen? Why did that happen? And that's what we're going to talk about here today. So, melanocortin-1 receptor, or MC1R, is a hormone receptor, and it is associated with color differences in mammals. So, it's basically how we have color changes, and it's located within melanocytes, which are usually inside of hair follicles. And so it is monumental in the biosynthetic pathway that creates eumelanin, and eumelanin is basically a color pigmentation. And so down below we have on the left side is a MC1R diagram, and so this particular diagram is showing just different mutations and how they could be damaging. But what's important to note is that the MC1R has an extracellular and an intracellular area. And the intracellular area is actually where the mutation occurs. And it occurs in 199, which is towards the end, which you'll see in my 3D model. And then, basically zooming into that is this second picture here. So you can see how the MC1R has two different pathways it can take. It can either bond to the alpha MSH or it can bond to the Agiyuto pathway. And usually when it bonds to the alpha MSH, that's when we get darker pigmentation, because it goes to the alpha MSH and then it goes through the pathway to create eumelanin, which is produced in the melanocyte. And from the melanocyte, which is in this third picture, it goes from the melanocyte, which is in the hair follicles, up the hair shaft to the hair cortex where it is shown, where it is seen. This particular mutation is called an SNP, and an SNP stands for a single nucleotide polymorphism. So that means that only one nucleotide is changed out. So in the unmutated version of this, we have the arginine side chain. And arginine is the original, amino acid that's used, and its side chain is called the guanidino group. The guanidino group bonds to aromatics and aliphatics above and below the guanidamine plane, but it bonds to polar groups within the plane. So it can bond to both nonpolar and polar groups. And this group is important because this is what would bond to the group that starts the eumelanin generation. However, in mutations, the cysteine side group, which is a thiol, is what comes into play. And so thiols are nucleophiles, and they're highly reactive, and they don't react well with the alpha MSH. So that's why when this mutation occurs, there is no eumelanin generation. And so how this affects functionality is that it makes this mutation makes it more difficult for the protein to bond to the alpha melanocyte, which is the alpha MSH, and producing melanin, so it makes a lighter pigment. This right here is my 3D model. This here is my 3D model of the MC1R protein. And the actual model, the actual version of this would actually have amino acids throughout the entire thing. It would be less uniform, and these red sections don't actually denote any difference between, like what the amino acids are. Those are just the only places I could make there be a more three-dimensional structure. So, other than that, this is, the most complicated model that I could make with this software. And right here is the location of the SNP. So right now it is being represented as being arginine 67, but once the color is changed, it is now representative of being cysteine 67. And this can show how simplistic a mutation it is, but it is still monumental in changing whether or not melanin is produced. And this is how the cell looks when you go from all angles.